All right, we are back. I'm here in the classroom recording my little blog videos here on Tuesday morning, and which I didn't get done, of course, yesterday. Sorry about that, guys, but we'll get them up now. All right, so what we have left when we were talking about the cell, we have a few of the organelles that we need to talk about, and one of them, that one. I've drawn a picture of it on the board over my shoulder. Hopefully you can see it good. It is a mitochondria. And when we talk about the cell, we had talked about all this work that it was doing. We were saying it's making these molecules of protein, and it's detoxing. We talked about all this kind of stuff, eating stuff, that requires energy. So it requires energy, and the energy that the cell uses is not, it has to be converted into a form that is usable by the cell. So you eat a Snickers bar or carbohydrate load, eating, maybe you're going to run a marathon, eating spaghetti and high carbohydrate foods, but at the cellular level, what has to happen is that macromolecule, that molecule of sugar has to be, or protein or fat, has to be torn apart, broken down into individual carbons. So you have this macromolecule that has to be broken down into individual carbons, releasing the stored energy in it. And that stored energy has to be used to build ATP. And we said ATP is the form of energy that can be used by the cell. And the organelle where that happens is this thing I've drawn over my shoulder here, the mitochondria. And on your slides, you have a picture from a micrograph where they've highlighted it with yellow and orange, and then a little sort of a cartoon type drawing. And there's all these pieces and parts of the mitochondria. And so we're going to learn them now, but you're going to use this understanding later this semester when we talk in a little more detail about this process of aerobic respiration. That is the process of converting the energy stored in macromolecules into cellular usable energy, ATP. And so when we look at this diagram of a mitochondria, you can see it has an outer membrane, and then it has this inner membrane that has its own name. It's called a cristae. And in the very middle of this cristae, the middle of that mitochondria, we're going to call that the matrix. Sounds like a movie, doesn't it? It's a little different. The matrix, it's that interior. And then we have a space that's between these two membranes called the intermembrane space. Inter meaning between. So what a crazy organelle, right? It's why on earth would you have this double membrane? And scientists have pondered that very question. And what they realized, and I think I mentioned this before, it's confusing to me because I teach two classes that kind of parallel each other. But if you think of an amoeba, I keep talking about amoebas or white blood cells. If you think of a phagocytic organism oozing along and eating stuff, what does eating even mean? It means that you engulf something and put it in a second membrane. It's pulled into a ball, which is made of cell membrane, right? It's a vesicle. And we would call that a phagosome, which would be a food particle in a vesicle. And what did we say typically would happen? The lysosome would bubble across, join up with it, and digest it. But what if you were a bacterium and you could do all this marvelous stuff with sugars, making ATP, and you were willing to share? You needed some ATP, but you could make way more. It might be kind of a good thing to have in your cell. And so throughout history, this is we have this theory, another theory, called the endosymbiont theory that is trying to explain this thing. There's one other structure in here I haven't mentioned yet. DNA. Not human-like DNA, bacterial or microbial type DNA. So this thing really looks like it was a living creature, a single membraned living creature that was really good at converting sugars and other biomolecules into cellular usable energy. And when the cell ate this thing, and for whatever reason it didn't digest it, it gave the cell a huge advantage. We're talking about in evolutionary history. So it persists, and this thing, whenever we talk about cell division, cells splitting in two, these things do the same thing. They're inside of you making more of themselves prior to cell division. So there's a lot of reasons to believe that these were free-living microbial type organisms that somehow ended up inside of our cells in what we would refer to as a symbiotic relationship. And that is a relationship where the lives of both organisms are linked together. They can't survive at this point on their own. And you and I, if you have a metabolic disorder where you lose your mitochondria, you're in really bad shape. So that's how we think we got mitochondria. Just as a quick aside, we're not learning anything about plants in this class, but if you know, the green parts of a plant are what we call chloroplast, and they are a double membrane organelle with its own DNA. It's thought to originate in the same manner. All right, enough for that. I'm gonna move ahead with one more organelle, and that'll be a really short video, I guess, or maybe I'll just tag it on here, and it's something called the centrosome. Now, way back when, in the previous set of videos, I talked about microtubules. And we had said the microtubules are going to be holding stuff in place and acting as pathways around in the cell. Well, whenever the cell undergoes cell division, you have to get all of that DNA, you make copies of all of those 46 chromosomes, and so you got to get one set to one side of the cell and the other set to the other side of the cell, so that when the cell divides, both new cells have complete sets of the DNA. Now, how is this done? It's done with microtubules, and it's controlled by an organelle called the centrosome. And the centrosome consists of two centrioles, and these are little barrel-shaped, look at the picture on the next slide, two little barrel-shaped structures that are used to pull the chromosomes to either end of the cell using what we call spindle fibers. They're microtubule, pulling them apart to either end of the cell, so when the cell divides, both of the new cells each have a complete set of chromosomes. All right, I'm going to stop right here.